Okay, today I will ask Shran, our TA, to come here to show you how to use 10x Genomics cloud service to upload FastQ files you get from the sequencer, from the single-cell RNA-seq library, and then map to the reference genome, and then download this gene expression matrix file. So this is from a raw data to matrix is a necessary step before we can work on the matrix of gene expression for the downstream. So everything I showed you last week, we start from the matrix itself, but how can we get from the raw data to the matrix? That Shran will going to show you using 10x Genomics solution. But before that, I want to make you be aware that 10x Genomics is one of the dominant company provide single-cell RNA-seq solution, but it's not the only one. It has several competitors. One is called Pass Bioscience. By the way, the market share should be 90% 10x Genomics solution right now, but gradually there are other companies try to catch up to provide alternative solution. So I provide some of this web page I will post them into the channel so you have some reference later. So this one is called Pass Bioscience. They provide a single-cell sequencing solution, which they claim their technology does not require this controller. The controller itself, you have to have this special equipment in your lab or in the sequence center to prepare single-cell RNA sequence library for you. You see the draw plate, the barcoding, all the system costs money. And their solution based on the, let's see. So they claim there's no instrument required, but you need to have their plate, 96 plate to prepare. This plate are specially prepared. Each wall contains different unique barcoding. So you split your cell population into 96 wall and then transfer to another 96 after mixture and transfer another. Every round you barcode individual cells with a different barcoding based on the plate you have been given. So you see each of the cell has been labeled with a series of different barcodes. Eventually, you should be able to uniquely label every mRNA from the same cell using same combinations of these barcodes. So that's one of the competitor of the 10x Genomics. It's called Pass Bioscience. My lab used this technology before, but although they claim there's no equipment required, but still the labor cost or the training for the technicians, you need to learn how to transfer mixed cell and transfer to another plate. This also a procedure will take 2 days. So it's not free and also require some human intervention. So you need to be technically strong enough to prepare this. Well, 10x Genomics, you can just give the fresh cell to the controller. Controller will make droplet for you. So no technique really required. Another progress is Illumina. So Illumina acquired a startup company called Fluent Bioscience. So Fluent Bioscience also have a solution which do not require does not require the equipment, no instrument to prepare single-cell library sequencing. So 6 months ago, Illumina acquired this company and make the product. So this is the news on everywhere, and try to make their own single-cell RNA-seq solution. And 1 week ago, and they released this. Okay, so Illumina single-cell 3' RNA sequencing preparation. So this is head-to-head competitor to 10x Genomics. And they claim their solution is based on the Fluent Bioscience, the company they acquired 6 months ago. But now their solution is this, and also 5 times cheaper than 10x Genomics. No instrument required. It's involved in the, let's see, 15 hours, okay, 10 minutes and 100. So similar numbers of these outputs, and the scalable, flexible, high quality. So that's a procedure involved. Sample preparation, you just need a vortex mixture. So you just basically shake your cells and let it to mix with I don't know, they are the template they provided, then every cell will be labeled. And because you know Illumina is a sequencing provider, so they have their own sequencer straight to from the library prepared to there. So there's some advantage that the 10x Genomics may have a very strong competitor right now on the market. But this doesn't create a big news for some reason. I don't know why, but to me, if they can reduce the cost by 5 times and also reach to the same quality, we have to test, and then that will be the big news. So I gave you this idea is the 10x Genomics is one of the most dominant and mature technology on the market right now, but it's a changing world. Maybe one day we use something else to replace. But the principle is largely the same. You have to have some barcode system to label the mRNA from individual cells, and then bioinformatically, then you can separate those rates and partition them into individual cells. Okay, so I have a question for you: is are you all understand the mapping? So when I see map short rates to the genome, do you know what that means? This is the basic knowledge for the next generation sequencing data analysis. When I see mapping, if you know what I mean, raise your hand. 2. MAPPING. Mapping. Okay. Sorry about that. So basically, you have short rates, and if you assume the from mRNA from one of the genes in the genome, so you want to find out which gene this mRNA is transcribed from. So you map this to the genome by look at the DNA sequence and find the consensus piecement fragment, and you map that to the corresponding genes. If you one of the gene map, there are many rates map to the same gene. That gene expression level is relatively higher than others. Okay. Okay, so I send out before that, I want to make sure you by the way, if you do not know what mapping is, Sharon is here, and email him and make appointment. And also, most important, everybody need to understand this slide. If you don't know how to explain this, email us, ask Shran. Okay, why this work? Okay, what's the GeoBead? What's the barcoding? The barcode from text. What's the UMI? Okay, yeah. Yeah, so this barcode, this is the green one. It's all the same. Yeah, consistent across this GeoBead, but it will be different from another GeoBead. Well, UMI will be, this fragment will be different. So although for this GeoBead, this droplet, you have all the same barcode, but each of these UMI is unique. So you can consider this like a hairball. There are a lot of hairs, and then the base of the hair, the green ones are all the same, but the tips of the hair are all different. The different ones will capture each individual mRNA molecule in that cell. So if you make multiple copy of the UMI, you will be eventually counted only by one, because that's not one single capture of mRNA. You made PCR duplicates. That's what I call UMI. So okay. So if you still have any doubt in this, email us, make appointment in person or online. Let Shran to explain it to you. You have to be very, very clear what that means, each of this. Okay. Now I will invite Shran to come here, and he will provide example FASTQ file. How many? 2, should 3? 2 files.
One FASTQ file, 2 reads.
Okay. Yeah.
It
Yeah.
should be on the Teams. If you all are logged in on Teams on this machine, I don't know, but I have sent a link there, and you will be able to find the files that I want you to download for this tutorial. Just let me know once you'll get the message. I can wait.
Yeah.
Get the message.
Let me see. I saw. Can I stop here?
Yeah.
Okay. Yeah. Let me close this, and then
Should be in general.
general. Yeah.
Yeah.
So in Teams, in this general, the channel, Shran just upload these 2 example. It's on your Notion. Yeah.
Yeah, it's a website, so people can access it. It has all the steps as well as the data set. So
Mhm.
I'm just gonna click here so people can.
Yeah. So you can use this.
Yeah, I think I can. Am I audible without the mic? Yeah, cool. I won't need the mic.
Okay.
Okay. Hi everyone. I am TAing this semester for Dr. Kai. I'm also a 3rd-year PhD at his lab. I'll set this up. That switch. I'll be just running through this tutorial and just getting you all familiar with what the Cell Ranger pipeline is and how you deal with the FASTQ files that the TIGS or the genomics core at A&M sends you.
Yeah. So you have to
Yeah.
mirror your. Already done that. How to use that?
Yeah, it's a little strange with the Mac.
No, you have to use this.
Which one?
The content over here. So you have source.
HTML.
Oh, HTML. Yeah, so you have to
Yeah,
prepare that.
it's
Okay.
let me just set this up. There we go. Okay. So the first thing that I want you all to do is create an account at this website over here, which is the 10x Cloud Analysis. Once you all are done with that, just let me know if you have had any issues with that. Shouldn't be much of a hassle. You can just sign in through your Google account as well. So this website basically allows you to run the Cell Ranger pipeline on the supercomputers or the high performance computers at the 10x facility. Otherwise, you'll have to make an account with the A&M High Performance Research Computing or the HPRC, which is, A&M can also run it. They also have the Cell Ranger pipeline. But this is easier because you can just drag and drop files and they will do the analysis for you. A&M, you'll have to code it on command line. I will also share at the end of the class the resources and the tutorials if you want to set it up on your local lab Linux machines or on the HPRC at A&M, how to get the reference genome and the mapping done. So while you're making the accounts for the Cell Ranger cloud, the cloud analysis for 10x, I would also like you all to download the files on this website over here, which is the 1K mouse kidney nuclei isolated from Chromium. These are all technical terms. I'm not going to get into that. Yeah. Yeah. There we go. Yeah. So this is going to lead you to this page over here, which is a repository of all FASTQ files and datasets that 10x just has on their website to advertise their technology. But we can use this for our analysis as well. So what I want you all to do is I want you all to download the input files and over here just the FASTQ files. It's about 3.5 gigs, so it's with A&M's internet speed, it should take 5 minutes to get it set up.
So how can we get into this page?
Oh, it's on there. It's on the Notions.
Yeah, that's just wait. So I will just download it to your own folder.
Yeah, just download it on anywhere in the downloads file. It's fine because we'll be uploading it back to the 10x Cloud.
Walk around and make sure everybody's on the same page.
Yeah, I can also go ahead.
So FASTQ file is what you will get from sequence filter. That's your role in nature.
Yeah, just keep the cloud analysis page open.
What is this I1?
There we go. Oh, okay.
I think this old version they have this index.
You might need to provide the
new version. We only have R1, R2, R1.
That's how they keep track.
That's a good point. So this may be even older. Sure, your data set has two R1, R1, R2.
Has to also
two index files.
Yeah, so it's a new technology. I'll come to that.
It's a new technology?
Yeah, so those are the indexing files for multiplexing.
Oh, I see.
We don't need that. We don't actually
We
need
don't
that.
actually need that.
Because this is just one sample, but when you're pooling in samples, they have now this. Yeah, so when you have the R1, R2 files, when they have pooled samples, they have these multiplexing or
New older.
This is a newer version. The most important files that you will be getting if you are doing sequencing ever after this is going to be the R1 and R2 files once you get there. Because those are the reads, the pair-ended reads from the 3' Yeah. Yeah. This should actually, if they run it, the data set will be prepared by the end of the class, so you can actually download.
This is we will download. We will map this
Yeah.
into mouse or human genome. I don't know.
Mouse.
This mouse data. So we then will download and input into the mathematics.
So this is a very small data set. I mean,
So today I think we can go through the whole thing from the
Yeah.
raw data to the final annotation of the sum.
we can run that.
Then we are almost done with the class.
Okay. So how's everyone doing? Everybody got the files? You good? I can move on. Great. Okay. So I'm just going to start off with what the cloud analysis is, what is mapping, and what it basically does. So just as Professor mentioned, mapping. What is mapping? So what the sequencer does is that when you're doing the sample prep, your cell contains a bunch of mRNA, and those mRNAs are mapped onto your reference genome. So for this case, I want you to concentrate. This B, D, and E are not really what we do for single cell, but I want you to concentrate on this, A and C. Each of these small mRNA pairs, mRNAs are mapped in this case from the, we don't map the whole mRNA, we just map the front ends of the mRNA and the back ends of the mRNA. Because these reads that we are mapping are only 250 base pairs. Recent technology has developed, and you can actually map the whole mRNA and actually see at the splice variance, that is how the introns and exons are cut off. But for the cheapest technology, and most probably what you will be running is going to be just these short read sequences, and that maps only the start of the mRNA and the ends of the tails of the mRNA and from both sides of the DNA. Sorry. Yeah, basically that. So now what we do is that we get the pair-end mapping from the end and the starts over here, and we get this regions, these short reads. Now we don't know where these short reads belong. So what 10x does is that it labels each of these short reads by the barcodes and the UMIs. Now the barcodes are same for each cell, basically, or each gel bead, because each gel bead is supposed to contain only one cell. So each gel bead has one barcode, and each molecule inside it, that is each mRNA inside it, has a unique UMI. And now, once we identify these, we can actually map it to the reference genome and see where these reads are actually mapping on the exact chromosome, or which chromosome and which distance, length, or position, I would say. Okay. Now, what the cell ranger pipeline does is that it performs alignment, it counts the number of barcodes, that is the number of cells, and it also counts the number of genes, particular genes in that cell. So thus it creates a matrix which has for the number of genes and what gene it is for each cell. And then it just filters out the empty barcodes. That means the gels which actually didn't capture a cell. So this is what the cell ranger pipeline does. And this is just a representation. I'm sure Professor has already gone through this picture. Not really. Okay, but I think he has it in one of his slides. So this one is what it exactly looks like. This over here is a unique barcode for each cell. So all the cells will have the same barcode in this case. And this UMI might belong to a particular gene. So the read from that particular gene. So if a cell has two of the same UMIs, that means that cell has 2 reads of this particular gene. And that's basically how the mapping works. For the mapping to work, what we need is basically the FASTQ files. The FASTQ files are the reads. That means the short 200 base pair reads from the mRNAs that was captured and then PCR amplified. Then we have the reference genome. The reference genome is the whole FASTA sequence of the whatever organism you're working with, basically human or mouse. The primary assembly, and you can find it on this website. On some, oh wait, that's a spelling error. There's no E at the end. On this website or the Ensemble website, you have the reference genome in the primary FASTA file, which is the whole DNA sequence, and the GTF file, which labels which region of that chromosome is which gene. Now, once you have all of this reference genome and the FASTQ files, you can input it into the Cell Ranger pipeline, and it's going to generate the count matrix for you. Now, the 10x Cloud pipeline has the reference genomes for human and mice. But if you're working with other animals, and you're in the vet school, I don't know how many of you are in the vet school, but you might be working with other genomes like cats, dogs, sheep, horses, whatever. Then you will actually have to create your own reference because the cloud analysis doesn't provide it by default. For that, you will, again, this is what you need, the primary assembly FASTA file and the GTF files. That thing, the creation of the reference genome can't be done by just clicking buttons. You will actually have to code for it. I have the tutorial for making a reference, and I've kept it at the end if anybody ever needs it in the future. Next, I'm gonna just explain and I've had a lot of collaborators ask me over the time, like, what does these numbers or things mean on the FASTQ files that you might be getting right now. So if you open up the FASTQ files that you just downloaded, you will find a tag at the start which says the 1K mouse kidney CNIK 3 prime version 3 I don't know, sample 1 lane 4 R1 is probably one of the ends of the parent sequencing. So the first part is only the sample name. This one you might not find in the TIGS, the files that TIGS generates, which is basically the technology that was used for creating the library and preparing our samples. So in this case, we are using the 3 prime version 3 technology from 10x. And this might be different. It might not be there. Now S1 is sample 1, and lane 1 is when you have the sequencer, you put your samples in lanes, or like these are wells which basically have your single cell emulsion in it. So you might have different lanes. Different lanes might have different samples, or you might have multiple samples pooled in lane 1 only. You have, like, just for example, you had 4 mice you were doing for experiment, but they were all just same biological replicates. So you can actually pool the data into one lane. So in that case, you will have all the data in just one lane. So you can look into these files and actually differentiate which sample is which, and so that you can keep a track and you don't mix up samples later on. And next, now you all have the files. So this is just something that I wanted to get clarified, and now we can get started with the Cell Ranger pipeline. So the first thing that I want you all to do is go into Cell Ranger and create a new project. Once you have made, just name it whatever at this point, maybe class 1. I have already created a new project, so I'm just going to give you all a minute to just create a new project on the cloud analysis website. Let me know if you all have any issues with that. Yeah, can you go to the project? Does everybody have the gzip or the WinRAR or something on the machine where you are able to unzip the files that you downloaded?
Right.
Yeah.
Here.
Extract it. Yeah, that works.
Every machine.
Every machine should have it. Yeah. I don't know, but maybe that machine didn't have it set up as default.
Yeah, so you have a file basically the several files that you come put together, then you have to do extract it, and it will be separate.
Yeah, you don't have to download from us. But if you are downloading, can you scroll down and go to
Yes, sir.
but you don't need to do it. You have already have it set up. Is it a specific kind of mouse? Oh. Okay. Yeah. Yeah, that's what I'm wondering. Version 1, was it 3?
Do you have a team? In general, that's the link.
Yeah. 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 So that's what you do. But you don't have to do it for us. Whatever if you want to do for other animals.
Quick question. If I can't unzip on WinRAR, what other software can I unzip?
You have to send it, right?
I think I extracted, but I think you can. That has to do with the change that key again.
Yeah, it's fine. You don't need to
Okay.
unzip this.
Okay.
You don't need to. Okay, just do not unzip the gzip files or the .gz. Oh, you got to. Yeah, I know. Cloud and also, you don't have information. Just give me the next one.
Yeah, so that's your own machine. You don't have a is this universal machine? Okay.
Yeah. Yeah. Yeah, so you need, just keep it unzipped. Just check. If the files are highlighted. Do not unzip the fastq.gz files.
Yeah.
Just unzip the main folder. Linux will take in fastq.gz file format or gzip file format. No, wait, where is it? Oh, you need to unzip this. So where does it go? Oh, actually go down. Yeah, we have that. Right? Yeah. Yeah, that's the folder we'll be working.
That's the one that was, I didn't do that one, I was using it, so it's fine. It was like from.
That doesn't make any sense. Can you open that? Oh, maybe it's fine, that's the folder we. It says 2022, but I think because the data set is from 2020. Like after you unzip it, the date shows 2022, right?
Oh, okay.
Because I think the 10x made the data set in 2022 and somehow that date has been preserved. That's strange. But anyways, so once you all have that file unzipped and you have 4 files in it, you can upload just the FASTQ files, which are the R1, 001 and R2. Sorry. Yeah. I don't know how to increase the font of this, but yeah. So I want you all to upload by clicking on this button, upload files, browse files, and upload just R1 and R2, both of them. And now this is extremely important. You always need to keep the R1 and R2 files together. So you upload them together. Now, after you have uploaded them, 10x is actually going to squish them into just one file, because those two files are just the two ends of the same mRNA that it's reading in. And this is just going to take a couple of minutes.
One sample. If you have two samples, then you have
four R1 and four files, a pair each. I can actually show the cell annotation. I was playing around with it yesterday. 10x has actually gotten the cell annotation inbuilt to it with the human, or the mouse, no, the human genome. It's not that great, but it gives you cell types right off the bus. Yeah. But I would say it's not that good. It's not good at all. It's very vague.
But the information is stored in the H5 file?
No, you have a separate CSV file with it. Okay, this should, once you have started the upload, this should be done in about 5 minutes. These are not big files at all. Like when you run your own sequence, you might get files which are 10 to 12 gigs. That's going to create a lot of issues. Yeah.
I have uploaded all the 4 files. When do you do it?
If you uploaded the 4 files, it's fine. I can, yeah. But that's just the newer technology. So if you get 4 files, you upload them all. You're not on 10x cloud. Wait, what? Can you go back?
You're looking for the file. Oh, you already have it.
Oh, yeah.
No problem. Yes.
I'm trying to figure out how to split cookie. I downloaded this, all of like this, into not the file that I was literally showing you, just this thing. So I'm trying to figure out if I should put it back really quickly. It was this one. So I was like, should I put this into this or just
Doesn't matter. As long as you know.
It's just like said
You can put
word.
it when it's done.
You drag into
That way it's all in one.
You would not have asked this last year. Yeah.
But I think that's still uploading.
Oh,
The machine is just still reading that, so it cannot read.
okay. Maybe after class.
Yeah.
That should get done. What?
No, I was just curious if there's an FTP protocol for 10x with the cloud.
They do. They have a CLI. You can do an FTP protocol with that.
Okay.
So they have a cloud interface manager.
I mean, at that point,
You need to
just use
run on this.
easier.
You see this button here?
Yeah.
Yeah, that's you need to upload, install the CLI on your Linux machine, and then you can just transfer files up and down.
Yeah, for the real world project, we spend a lot of time on this, just wait for uploading. Sometimes we don't know there's one file crashed in the middle,
Yeah, so if you have the HPRC, once you've created a project, you can use any of these commands to start the uploads.
But can I upload it from what I have already, that cluster, or no?
Yeah. You don't have to download it on your local machine.
Yes.
So you will be using this thing over here, the txg file upload, and the txg, and this will be the path to the file. You can copy this link over here. It already has your project token over here, which is the project that you just created. So it's going to upload the files only in the project folder. And the txg over here, you can click on this link right here. So yeah, so you need to set this up over here and then extract this txg file. This txg is the one that allows you to transfer files directly through the FTP protocol from any Linux machine, which is basically an HPRC, as long as it's connected to the internet. Okay, so once everybody has uploaded it, it should pop up something like this with the R1 and R2 together. That means that these are the correct paired end files, and they have been squished into just one file. Yeah, so I'm gonna tell you how to start the analysis, and you can. Oh, just click on upload file. Upload files. Yeah, click on upload.
Click upload file, and you'll see like
Yeah. So you have all the commands and the token, which is basically used to identify which project you are uploading to. That's how you can set it up. Yeah, what's up?
out of curiosity, for the virtual machine, is there any difference in having the whole exon versus the whole genome?
Yeah, it depends if your mRNA is not. So it depends if you want to do velocity analysis after this. Now velocity analysis is not been done here as of now, but that is a way of single cell analysis where you are mapping the splice, the unspliced RNA versus the spliced RNA, so that you know that which cells are actively producing RNA, so they will have the unspliced, they will have a higher number of unspliced RNAs. And yeah, if you don't care about spliced and unspliced RNA, it should make a difference. Is it good, Professor?
Yeah.
Okay. So the next step is setting which library or technology type this particular file is. So you need, so this is a 3' sequencing, as you can see from this 3P over here that I had mentioned. So this one is the technology that was used for sequencing these files. So I'm going to use the universal 3', and this is gene expression, and this is version 3 over here. So I'm going to set it, this library as 3' gene expression version 3. And your samples might be something different, so you'll have to set it accordingly when you are doing your single cell. And just after setting that, has everybody set it up as next gem 3' gene expression version 3? Yeah, cool. So you just take this file over here and you create a new analysis. You keep this as same, which is the most latest version of Cell Ranger. Give it a name. I'm going to just give it test2. And this is the most important. You need to select the correct reference genome in this case. Ours is I've seen a lot of people use human on mice sample, and that just destroys the whole mapping. Or if you use mice on human sample, it just destroys the whole mapping. So our samples are mouse, so I'm going to use mouse.
Slow down.
Yeah.
Whatever name you want to do, like so which one to recommend?
Mouse. Mouse, the most recent one, which is GRCM 39, 2024A. So that was the 2024 update on the mouse genome, as far as I remember. They still don't have the new T2T human genome in there. But anyways. Now, if you want to generate BAM files, which BAM files basically contain the whole FASTA file with all the reads actually mapped onto them. I don't need the BAM files for this analysis, but so I'm going to turn it off because those are extremely heavy files. If you have, you can just imagine, it is the whole reference genome with all the reads, which was already a 3.5 gig file. The reference genome is another 1 gig file, so it's going to add up. And it's going to just, you can just add them up. It's going to be around 5 gigs. So if you need the BAM files, a lot of people preserve the BAM files because they need it for when they're publishing the paper, they upload the BAM files along with the paper, but just for this case, I'm turning it off.
Some analysis do need BAM files.
Yes.
For example, RNA velocity. It will require you to figure out the splicing site. In order to count spliced and unspliced mRNAs. In that case, we do need BAM files.
Yeah.
In general, we just count how many copies of that mRNA expressed for this gene. But in that case, in our simple case, we don't need that.
But if you are doing,
print off, we just save some space when we download.
we are just saving some space at this moment, but I would recommend keeping it on because when you're doing your actual analysis. And you're going to run analysis, and it's going to run it by itself. And it'll email you once it's done. And this is going to take about 30 minutes. So that will be each.
So just to make sure everybody submits the mapping job that the machine can run on the cloud until we get the output file. Then we will look at what the output looks like, and we download and put it in the lab. So this is one sample. If you have another sample named differently, then if you repeat the same thing, you download another sample data. So one matrix, another, maybe you have a third, each independent from each other. Okay.
Just let me know if you all have sent the file, submitted the file. 1, 8 this time. You're good. You're good.
I think Sarah may need some help. She's still uploading the file. Almost finished.
It prepares it. It's
So you can do this on your Linux machine. You can install Cell Ranger yourself and use command line to achieve exactly the same thing. But nowadays, even ourselves, we just upload to it. Even though it takes some time to upload the file, it's still okay. And as long as we get output, it saves us support. sequencing of library, and each library is sending you two files, R1,
The FASTQ files.
Oh, yeah. So it's basically like, so what's to be done on our part is like the single cell suspension, and then they give us the files for this.
Yeah, so you gave the tube with your suspended cell isolated, nothing, the fresh cells basically, blood cells you just give to the library preparation, and then you run genomic controller, and then you send the library to the sequencing center. Sequencing center send you a link and download FASTQ. So you output your tube with the cell, fresh cell. Your input, you input back is the FASTQ file.
Okay. And that's why, like, we'll have these files like 250 samples.
Yeah, pretty much.
And what are the other two files?
Those are when you're pooling in samples, when I mentioned that. The lanes, they're going to have the same lanes, but you have like 4 biological replicates you want to save some money on the sequencing. So you don't run them on 4 different lanes. Instead, you squish all of those cells into one suspension and give them as one sample. But that one sample actually has cells from 4 different mice per se.
Okay.
So that information, those 4 different mice were actually tagged differently when the suspension was prepared.
Yeah.
So you can keep a track.
On which cell is
Which cell came from which mice.
That makes sense. Okay.
So that's when they generate a second layer on top of that, the other 2 files. That's a fairly recent technology. I think it just came in the past couple of years.
So just saves money because you're able to like identify cell lineage.
Yeah. So it's not going to save you money for the sample prep, but it will save you money on the sequencing.
Yeah. Okay. Thank you.
Okay, so this is going to take some time to finish, and I'm just going to go through what the output is going to look like. I have already run the same samples, and this is the QC report and the cell type annotations that they generated automatically. And these are the files that they are going to generate for you. It's going to be a Lupe browser file. This Lupe browser file is basically 10x Genomics browser application for looking into the 10x data. And this summary HTML basically has all the information on how well the reads were mapped, how many reads were mapped, and what is the quality of cells that you have in there, like the total number of genes per cell and all that overview. And this is what we'll be working with, which is the feature cell matrix filtered. This filtered file has already removed the barcodes, which did not have any genes in it, basically the empty droplets in our case. So I'm going to just click on the summary HTML. Summary HTML is basically the same thing as this QC report. Yeah. So in our samples, the analysis estimates about 13,000 cells with a median of 2,500 genes per cell and 27,000 reads per cell. And this is just information about all the chemistry that was used for sample prep, the transcriptome that it was mapped to. So if anybody sends you all files that are already mapped, you can keep a track of what happened in the analysis using this web summary right here. In this case, out of all the barcodes that it detected, 98.2% of them were valid barcodes. That means they were not empty. And all of the UMIs that were detected were mapped onto something. And these numbers might be lower for your samples. This is 10x's ideal scenario, which they have prepared. So this is a good quality sequencing data. And these Q30 bases are just quality scores which are associated with the FASTQ files. So the FASTQ files have the sequence and the quality scores on it. So Q30 is a measure of how confidently the sequencer actually sequenced the mRNAs. So 93% of them are above Q30. I forgot what Q30 means. It's a confidence, like we are 99.999% sure that that particular nucleotide is actually A or a T And these are the number of reads that were actually mapped onto the genome. Out of all the reads that were read in, 95.9% were mapped to the genome, and out of which 93.9 were the ones that were confidently mapped to the genome. So this is what we'll get, or we only get these 93.9%, the confidently mapped genomes. And talking about the introns and exons, this is where the information is set in here. 52.2% of the reads were actually mapped onto intronic regions, and 36% were mapped onto the exonic regions. Now, if you remove the introns from the data, you won't get half these reads over here. And okay, that's basically it. These are the estimated number of cells. We already discussed this, and the mean reads per cell. It's already mentioned over here. And they already generate a TSNE plot for you all, so that you can just have an overview. And this is our gene expression data. We should get something similar when we actually get the files out and import it onto the MATLAB software. Something new that they have added is this cell type annotation result summary. This has been added. This is a new development this year. What happened? There we go. Yeah, this is the most recent update onto the Cell Ranger software. You can click on it and you can see what cells you have in your data set. So these cells were taken from mouse kidney. It says epithelial cell because it probably has all the kidney epithelial cell in it. But as I said, this is a very generic cell type annotation that they prepare. You can have it, you can look at it to guide yourself into what you might be expecting, but you will still have to do the cell type annotation on yourself on the MATLAB software. So it does put it into these subtypes that even within the epithelial cells, you have the distal convoluted tubule, proximal tubule, loop of Henle, connecting tubule, and so this is the fraction inside the epithelial cells. Endothelial cells also you have some of the, I don't know why they have. Oh, so these are the lymphatic endothelial cells, and these are the blood vessels, basically, and these are the lymph vessels. So they have subtypes for each epithelial cell over here. Now, this is what I don't understand. It says cell. So their automatic annotation is still not as great, but it's improving by the day as the human cell atlas improves, because they map it back to all the cells in the human cell atlas. But yeah, so these are the files that you will be getting, and this is a TSNE of the same representation. But we'll be doing all of this on the MATLAB software. We'll just be taking out our H5 files. Okay. So the previous, the one that I actually tested out, it finished in 30 minutes. So we'll just have to wait for this to reach 30 minutes or 35 minutes, and it should finish. Then you can port it into MATLAB. Yeah, what's up?
Can you grab how the rank score plot in QC results works?
Which one?
So in QC results.
Yeah. This?
Yeah, that barcode rank plot.
Oh, okay. So when this is the barcodes which are sorted by the UMI counts.
Right.
So if you look at this point over here, about near 1356 or 1364, that is the number of cell estimates. So whenever the number of UMI counts per barcode falls below 1,000, it filters it out. So it gives you this, it gives you two files. If you go over here, which is the feature cell matrix filtered and the feature cell matrix raw. The raw files actually contain all of these barcodes in this region as well. So it's going to have about 12,000 or even more. This scale is actually an exponential scale. It goes from 100,000, 10,000, and it increases. So it's going to have all the empty droplets over here. So whenever it falls below about 1,000 UMI counts per cell, it filters them out. That's basically it.
Yeah, we can take a 5-minute break. And you want to take a break, 5 minutes? Okay.
Because we have nothing to do at this point. I will go. I actually have a class at 11:30.
Okay, so you can. Thank you very much.
It's actually just the next class right there. It's Dr. Siberry's course.
Okay, sure. I had another class to take in
But I will send you the filtered H5 files.
Okay.
So that you, and they should be also getting the same filtered H5 files so that you can run it on the MATLAB.
Good job, Ashwin.
So hard to work with Mac at times. It's just, there we go. I saw because this was the smallest data set I could find on the 10x website.
Yeah, it's good.
So
Yeah.
I think that they can use the same data set if they want.
Mm-hmm.
I've sent it to you on Teams. That should be
Yeah.
the filtered
Good,
feature
good.
barcode matrix.
Yeah, that's what I'm going to do next. Good. So should I switch this?
where you are waiting for the analysis finish, I can spend more time on this QC summary report. That will be the one once you finish the mapping and get back the results you want to first check. Okay. I'm using one of the example from my previous analysis. So the number will definitely different from what you will get from this 1,000 mouse kidney cells. So this sample on my under my account estimate get 9,000 cells. And in each cell, there are 2,500 genes has been identified. This as a medium number. So in each cell, I will have a gene which contains non-zero rates of UMI information for 2,500 cells, genes. Some cells may be less, some may be more. Depends on the cell type, the quality of the sequence, or a lot of stochastic factors. But the medium supposed to be some of this level. If your medium is too low, that means you may not sequence deep enough. Maybe the quality of your droplet, the cells in there, not good enough. If this number is really high, I don't know, maybe you have a lot of duplets, cells, maybe super active transcripts. But for the general, we see this number, okay? Then this is how deep your rates have been mapped into each cell. So this also is a reasonable number. So you have some, based on the experience before, you should have 25,000 rates on average per cell, okay? So this number is much larger than that because some genes have thousands of copies. So you detect one gene may have 100, another gene may have 500 rates. Some gene may be lowly expressed, only have one rates. So the total number of the rates per cell is larger than the gene number. Of course, it's not one gene, one copy, not one rates. Okay, so this is three numbers here. Then you have to move to this part, plot here. This shows you accumulated UMI count number. So this is 1 to 1 million. There's nothing. This is just index. So this is barcode. Each barcode is one droplet. You know, we have nearly 1 million droplet in this library preparation. And those gray droplet are considered to be empty. Why? Because if you look at this droplet number, I don't know what the index is, the UMI count is really low. Okay? You don't expect you have a cell in there, then you get UMI count so low. So a lot of those droplets are empty ones. But once you have enough UMI count, you see those are the non-empty. There are cells in there. But we need to set a cutoff. So this is a beautiful curve because you have a sudden drop. Basically tells you your droplet clearly separated into two groups, empty and non-empty. If you have a not like this clear knee click shape, if you have a continuously dropped line and very smooth, that's not good. That indicate you have a lot of droplets, is half empty, half contains enough mRNA. So you don't have a clear cutoff. See where I should consider this droplet is empty. But in this case, it's very clear. It is a sharp jump. So we can set up a cutoff in this very narrow region. And I think by default, Cell Ranger set up the cutoff here, and anything above maybe 500 UMI to be considered non-empty. That's okay. You can change the parameter if you know how to adjust that, but normally we just accept this by default. We can do a better job downstream. We can set up a cutoff. We will throw away all the cells which does not have enough UMI. I will set a cutoff later in MATLAB. See, I don't want the potential empty droplet in this region. So I can do it later, doesn't matter. So this is the plot, how to read it. And those just count how many droplets, okay? So basically this number will give you that, okay? So if you draw a line from here and count how many cells, and how many non-empty droplet, that will be the 9,000, close to 10,000 here. Any questions about this? 3 numbers and this figure? Yeah, so if you finish your analysis, you should look at your QC and see whether you get this beautiful sudden drop of this this curve plot, okay? Then we look at some numbers here, and you have sample ID, and then which chemical, the version of the library, preparation chemicals, whether you included introns in the mapping, what's the reference genome and annotation you use. This is a human, a mouse, okay? And which version of the Cell Ranger you use to run this mapping, the number of reads, total reads, and all this valid barcode, okay? So tell you how the barcode system worked, 95% valid, that's good number. Let's see. So this report probably slightly different from the new version because they are upgrading the Cell Ranger pipelines. Before there's no annotation of a cell type, but now they have. Some mapping quality here, 95% map to the genome. So this is a good number. If you only see 54%, you may suspect that you map to the wrong reference genome. Human mouse may be okay. So if you map the human genome sequence into the mouse genome, you still get 85%. But if you map to another species, maybe only 45, something like that. So make sure you map to this number is big enough. There are in this case 61 mapped into the annotated transcripts. Okay. Yeah. Threshold, you mean all these numbers?
For the reads up to the genome at least?
there's no strict cutoff. Say, okay, I need to be in this or that. It's have to be determined case by case, and those numbers changed quite a lot. And also, this sample may have although the same batch, same experiment, but you may have different numbers for different samples. Okay, you know clearly this sample I have a high quality, and this, although I did the same thing but I get a low quality. It's always happen. But in general, this is very stable results, and you can make the data set comparable even though the experiment was done in different batches, in different labs, even in different chemicals. So far, in general, we realize the results are stable, very, very stable. Yeah. So that's why 10x genomics is still the number one. Okay, have you finished your mapping job? How many you finished? Not yet. Still. Probably have to download the file. Sharon just send it to us. Let's see. Yes, once your mapping finished, you should have results. Looks like this, right? We will browse down to the file that we really need. So the mapping, the result will be in this. Okay. As Fran explained it, this is the matrix contains only the non-empty droplet, the non-empty cells. And this one contains all, but so the file size is bigger, but we only need those non-empty ones because the cutoff already set up very relaxed. So this should contain enough information. So that's the only file we need from this particular sample after mapping. We need to download it and save to the local hard drive. Then we are going to read it into the MATLAB to do downstream analysis. A previous version of the Cell Ranger gave you 3 files, contains matrix, gene name, and barcodes, but now they merge everything into this HDF file, H5 file, like common file type that to communicate through the different platform right now. So what you need to do is just check the HTML, the quality, the QC report file. So it just showed you with the curve. Then you need to just download this filtered cell matrix. Feature means gene. So you have gene, cell matrix, HDF5 file, and you download in browser. Let me see, I can. If you don't have that, I can just share with you the Shran just provided. Yeah, in Teams, I just uploaded these two output files. So you should get it if you finish your mapping on the cloud. You should have a same output. They all named the same, okay? You can download this into your local drive. And then once you're finished, let's come back to MATLAB. Make sure you know where you download the H5 file. Remember I showed you, you need to know the extension so that I can see what's the extension of this file. So it's hard to see from the back, but in my download folder, I have this H5 file in there. This file is zipped. You cannot open, use your Word or Notepad. You won't be able to see anything. So this is a binary files. So I know my download H5 file is in the folder of downloads. Then come back to the MATLAB. I need to update my MATLAB toolbox installation. So I copy this page again. You can search single cell GA toolbox and the quick installation. You should have this page. Copy this and come back to the same place, which is in the same working folder, and paste what you copied. So this comment will goes to the repository and download the latest version and unzipped in the same folder here. The older version will be overwritten. It doesn't matter. Everything is also updated. So you just do this and you get the latest version, because I keep updating basically weekly. So I suggest each time we use this installation to overwrite the old ones to get the latest update. So I got this updated, and then I start to use this tool, SCGA tool. Type that in. Okay, in this interface, you click the import data and then select 10x genomics H5 file, and then click OK, and then locate the H5 file. You just download and then read that in. It should give you the cell expression matrix of the cells into MATLAB. Can you see the option? Okay. So I download into this downloads, and also I hope it works. So this is the Shrun provide us. It's 1,300 cells. Nice. So you have to point to the right position. Oh, this is type SC, lowercase SCGA2, GG expression GEA2, and then you select import with the H5 file. But it's okay, the same thing. That's good. I think it should be H5. Yeah. Yeah. Good. That's cool. Yeah, so if you want to look at the languages, okay. But you do not have to. The interface is okay. Yeah, it's supposed to be. Yeah. Okay, from here, the first time you use this, you may not have an idea which button to click. And on the most top, let me change my mouse cursor. Yeah, on the top
most left, if you click there, there's 1, 2, 3, 4, 5 buttons at the landing panel to show you which button you should click first. If you click number 1, this button will change to the button that you should click first. So if I click this and it says, okay, this button allows you to filter genes or cells by the quality, basically. So you learned, okay, which button you should click first. So it's this filter button you're supposed to click first in order to do the filter of the, get rid of the low quality cell and the genes. And then after that, the next button you should click is this movie film-like button, which is here, okay? So after the QC, get rid of the empty genes, empty cells, and you want to do the embedding to move cells around to make them ready to be clustered into groups. So after that, the third button you should click is this one, 3 bubbles. This will help you to cluster those cells into isolated groups by label them. Then number 4 should be this button here to allow you to annotate the cells of each group. And number 5, so after you finish annotation of this individual sample, remember there's only one sample, later we will add more, and 3 or 4 or 5 to compare to each other. But right now, we just learn how to deal with this single sample data. Okay? From the filter, embedding, cluster, and annotation, and save. That's all. Okay? So you can click through these 5 buttons and finish off the annotation to save the results onto the hard drive here. Then we are done today. Okay? Let me know during the process you have any questions, and I will work around. Okay? If you think, okay, I don't want to learn any of this, you can click this the single click solution. This will be automatic. Create all the pipeline for you, and you just one click and wait, you will get the results. That's what I'm going to do. I'm going to just click this button and let the program to compute all the downstream. Okay. But I suggest you click each individual button separately so you basically know what's doing. There's some options in the middle to allow you to make changes.
For the embedding, it's asking the methods.
The different methods. So you can select Kisney or UMAP is most commonly used.
3D.
3D is good for visualization to manipulate. 2D is for publication.
If you click this one over these 5 buttons, right?
Yeah.
And is that like most common type of the users
That's
is
right.
going to use?
Yeah.
But if we learn more specific things, you go.
That's right. Yeah.
Okay.
Yeah. You have more control.
Okay.
Each step. Yeah. The different options.
Thank you.
Yeah. a screenshot and you easily fix most of this. It's good. You're done. Did you use one click button? You can save this to the method. And then you can save the hard drive. So in the last step, you can save either as a temporary variable, or you can save in the hard drive. So next time you can still load the information back exactly the same annotation as it's in the
So how does the software determine how to separate the cells? What point to group cells? I guess the question is,
yeah.
What is the scale based off of?
Yeah, so it's based on the position or the cluster.
Okay.
And so that's what we will learn in week 3, something like class 3, and many options.
So I asked
class
next
3 is that the machine learning is really good at.
I'll ask you next week.
there are some theory behind that.
Yeah.
Different options. So we will have to gradually learn those options. Yeah. So I changed this.
Yeah.
Buttons.
Honestly, I like the old school style better.
I can have that. Yeah, I can tell you of that option.
Yeah.
Like, well, when my computer decided to send this, I don't know. Mine?
Yeah.
I
The
did it.
results, yeah.
Well, like,
Yeah, with different because
I use KV, the number that he gave me. This text I used.
in general, you can have consistent results. I don't know, biological.
Well, not problem size. I just think of
Yeah, I don't know whether my annotation. So this is from the kidney cells. There's some nuance in there.
yeah.
Yeah.
Well, the one that I used in the field team, I used this one, and we used the I used to kind of like
Yeah.
fix something.
Yeah.
Yeah.
So you just try to work anything. Yeah, so what you can do is shut down and restart. MATLAB. Maybe that's over. I don't know. It should be. In general, it should be very. So any questions? No? It's easy, right? So our lecture theory will go through some details in after you click the button, what will happen, algorithm theory, and then what other potential options, why use TSNE, UMAP, why not use TCA. Yeah, so that we will go through a lot of questions you may have later, okay? it will be crystal clear and what's in there each step. Okay. By the way, we haven't touched on the comparison between samples yet. There are others, differential expression. So how can you compare? How can you compare mean and variability? How can you generate a gene regulatory network for each cell type? How the gene regulate each other, they're different from one cell type to another. Then if you have two samples, how are you going to compare the gene regulatory network from one sample with another for specifically given cell type? We have pseudo-time analysis. We will align cells from undifferentiated or differentiated or different states. There are many options later on, right? Okay, the cell-cell communication, how the the fibroblasts may have communication in those same cells by some ligand receptor signaling pathways. Yeah, many different potential options for the downstream. Okay, slowly review all the details week by week. Remember, there were 13 weeks to finish all the class. Okay. Okay, so here, if you look at this hidden button, when you highlight this line, you should see there are buttons you can select to rotate the figure. Okay, and this you can zoom in, zoom out. You can use this palm to drag the position of cells. In some of this annotation, you have keep labels. I will show you there. You can label individual cells. So this yellow one, you can click and put your cell type label to here, and you can drag. So give you a lot of flexibility to customize the figure here. You can brush to select a group. So you brush and select this group, and then in this group I have a neuron and some of these distal tubular cells. Then it's a mixed population. Then you can merge, so I can either merge them into the same cell type. So there's some annotation you can do. You can make this a separated sub-cell type by select this cell and make this into a new cell type. A new cell type. Some manual annotation to help you. So I will fix this problem later. Cell type. In the plots, you can see I want to generate a clean t-SNE plot. So this will give you a view that looks like this. Okay. The reason I put that is t-SNE UMAP are the tools for visualization. Basically, this X, Y, Z axis has no unit. So some reviewers, if they know the algorithm clearly, they will see you cannot label any numbers on this t-SNE UMAP because they are unit free. You shouldn't have that in there. some cases you want to have a clean plot like this with no label of x, y axis. So this will be ready for the publication. Then you can click this button here to send this to either PowerPoint or if you use SVG or JPEG version, then you should have that figure in screenshot and put in ready for publication. But of course, in this case, the 3D. Normally, we should have a 2D for publication purpose. Those are just some options you can explore. And if you see, I want to do this, let us know. We can build in some of the function in there. It's really slow. I think I need to mention when you save this, you have options. Yeah, my machine is freeze. Yeah, I just mentioned when you save this, okay. When you save this, you click this save button here, and then if you still want to work on that, but you can save this in the workspace. You can save expression matrix X and G, remember big X, small g, and you can save this variable SC itself, then you will see them in this workspace. And then you can process those. Using command line here, so you can see, okay, gene, remember the index, the top 5 genes in my list, so I can print them out in the command line window to manipulate this data. You can also save to MATLAB file, so this will be easy for next time you load. So if I select this MAT file, then I will give a name of this test file, test1.mat. So this is a native MATLAB data file. You see here, it's on your hard drive. Next time you can load that SCE back. You can also save this into R. So if you decide I'm going to transfer everything from MATLAB to R Surrey package, you should be able to export to R. And this is for Python, so this is for ScanPy. It's called anode data. The end with H5AD. Okay, they're all possible. I think the last one I want to show you is so next to this PowerPoint figure button is this small camera. If you click this, you should be able to make a MP4 video for this cell population with the rotation. It's good to share this MP4 file with your collaborators. Say, okay, this is my preliminary annotation of your cell type. Does this make sense to you? And they will give you feedback, and now you can have a communication with your collaborators. with this MP4 file. Okay, so it will be generated in here. All right, this is all for this week. I will see you.